Hello lab viewers. Last week we had made some progress in our testing of petri dishes, and this week we'll move on to hydrogen peroxide testing. So let's see what that looks like.
We made aliquots of our bacteria by mixing samples of luria broth with glycerol, and we froze them in about 40 Eppendorf tubes. And we use these to inoculate bottles of distilled water to create bacterial solutions to make plates.
We made bacterial solutions by taking 150 microliters of our bacterial aliquot and taking 100 microliters of our bacterial aliquot and putting them in 500 milliliters of water respectively. From these solution we made plates by placing 100 microliters of the aliquot and water solution and on 2 plates and 50 microliters of the aliquot solution and water on 50 microliters on 2 other plates. This plate is missing because we threw it away, I think. And then these 2 plates both had 50 and 100 on of the 150 aliquot and water dilution. The ones of the 150 dilution, although they should have had more bacterial colonies than the ones of the 100 microliter solution, they have less, which led us to believe that the problem here is that we did not agitate the bacteria. We must have forgotten to shake the bottle in which they were contained. Therefore, the bacteria was not evenly distributed throughout the water and no colonies were present. We just have kind of large clumps growing here. In the 100 microliter solution, however, we did shake it and we have lots of colonies growing here that are visible. We actually have too many. The best concentration we had was of the 50 microliters in the 100 microliter solution, and that produced 326 colonies when we counted all of them, and we're aiming for about 100, so we are going to redo the solutions for our next experiments when we start experimenting with hydrogen peroxide. Because the plate with 50 microliters from the 100 microliter solution worked best, we decided, except for there were too many plates, we decided to make a new solution with 75 microliters and 500 milliliters of water, and we will use 50 microliters of that solution just as we use 50 microliters from this solution on our new plates. Because we're using distilled water in our solutions, we also ran tests on our distilled water to ensure that there was no bacteria in them. This one has 100 microliters of distilled water, and there were no bacteria present, so we can infer that our distilled water was sterile.
So this is the process of setting up and spreading the plates for each sample. After you add some 50 microliters or 100 microliters, depending on what you're adding, you then spread it like Charlotte is right there. This makes sure the liquid is thoroughly dispersed, and to set up the plate, you just put 50 microliters into the middle like I did there, and it's ready for spreading.
For our next experiment, we plan to add hydrogen peroxide to a sample of bacteria to see how well it sterilizes it. Currently, we are incubating 6 plates. 2 of them just have bacteria and water, so there are positive control. We have 2 that have 1 milliliter of bacteria of our 75 microliter bacteria in 500 milliliter bottle. So 1 milliliter of that with 1 milliliter of 3% hydrogen peroxide, and then we have 1 milliliter of, again, that bacterial solution, but with 0.125 milliliters of 3% hydrogen peroxide and 0.875 milliliters of just water. So those are 2 different concentrations. One is a 2-fold dilution for hydrogen peroxide, one is a 16-fold dilution of hydrogen peroxide. So hopefully, if even the 16-fold kills all the bacteria, then we'll know we can go lower and see exactly how much hydrogen peroxide it takes to kill all the bacteria.